Hello everyone. So welcome all of you to the next class, that is the next video on anatomy of flowering plant, and today in this video we are going to discuss about the phloem. Okay, phloem, which is also known as bast, and it is the second complex permanent tissue of the vascular plants, which is mainly responsible for conduction of organic food. Okay, the phloem tissue, as like the xylem tissue, it consists of four different types of elements: the sieve tubes or the sieve cells, the companion cell or the albuminous cells, the phloem parenchyma, and the phloem fiber. Now these elements of the phloem tissue were first of all discovered by Hartig, and the term phloem was first coined by Nageli. Now, when further studies were done, then Heberland, he considered phloem and he studied about phloem and termed it as leptome. So, Heberland called phloem as leptome. Now, the first component of the phloem tissue, that is the sieve elements. Okay. So, what is actually the sieve elements? So, this middle portion, as you can see, this is actually the sieve elements. Here, the cells are mainly meant for conduction of substances. The sieve elements of the non-flowering plants are called sieve cells, whereas in flowering plant the same thing is known as the sieve tube elements. Okay, they are always arranged end to end so that they form a tubular channel. Long channel is formed, sieve tubes, for which it will be easier for conduction of the particular foods. Okay. Now, these particular cells in between, they have the sieve plates or the septa. These sieve plates, they have some pores which are known as sieve pores, and these pores help the substances to pass from one part to the other. Okay. Now, the sieve tubes elements, they are arranged, already saying that they are arranged end to end. So the substances, that is the mainly the protoplasm, that which can pass through the pores, and they form a continuous channel which is known as the protoplasmic strand or the connecting strand. Okay, the pores through which the protoplasm will enter, so they will definitely form a continuous strand. Now each pore contains some cylindric of callus. Okay, that is which is actually made up of unbranched beta-1,3 glucan, that is outside of the connecting strand. So outside the connecting strands, they consist of callus layer. In non-flowering plants, the sieve cells, they possess some pointed or tapering cells, and the length of each cell is about 1 to 40 millimeter. The sieve elements are living cells but are without nucleus and mitochondria in their mature cells. So when mature, mitochondria, nucleus, these types of organelles disappear. They have peripheral cytoplasm, and in between vacuoles are present. Okay, and nucleus is there. Yes, vacuoles are present, sorry. In dicots, a few other plants, small tubular, granular, crystalline substances are present. That particular structures are known as the P proteins. And in starch, the plastids, in dicots, the plastid stores starch, and in monocots, the plastids will store proteins. Okay, that means food that is prepared is stored in the form of either starch or proteins. Now, second is the companion cell. These are the helper cells which will assist in transportation of substances. They will help the sieve elements to work further. Okay, they are thin wall living cells and are associated with the sieve elements. It occurs in flowering plants and develop from the same mother cell from which these particular cells develop. So sieve elements and the companion cell, they develop from the same mother cell. Here the cells are nucleated and the cytoplasm are rich in ribosomes, mitochondria, plastid, endoplasmic reticulum, etc. Now their main function is conduction, plus they help to maintain the pressure gradient in the sieve tubes. Okay, and in non-flowering plants, these cells are associated with the thin wall living cells called the albuminous cells or the Strasburger cells. Okay. So these are the two elements. Firstly, this is the sieve tube element, and second element of phloem, that is the companion cell. Now we come to the third part, that is the phloem parenchyma. This is also another transverse section. Phloem parenchyma. The parenchyma name itself indicates that these are living cells. Okay, they are thin walled with dense cytoplasm. They store food, resin, mucilage, tannins and latex, and they take part in lateral conduction of food. They are normally absent in case of monocots and some herbaceous plant. The fourth element is the phloem or bast fiber. That means the sclerenchyma cells, which are associated with phloem tissue. They are elongated, dead and empty cells with pointed end walls. Okay, and these cells are thick lignified, so definitely they will provide mechanical support to the particular plant. Now, as similar to that of the xylem, the phloem is also again divided into protophlloem and metaphlloem. So, protophlloem means the part of the phloem in which the phloem is formed first. Okay, these are smaller sized, and metaphlloem means the phloem which is formed after protophlloem or from protophlloem, and the size is larger as compared to the protophlloem. Okay, metaphlloem are larger and protophlloem are smaller. Okay, so this is a simple portion. So the notes, the video and the diagrams are provided to you. Go through it properly. If you have any problem, then let me know.